Yep. We're a Zoom company.
Yep.
Awesome.
Yep.
Well, would you
All
like
right.
me to go ahead and
So let me go ahead and briefly introduce, and then
awesome.
it's all yours. Yes.
Yeah.
All right. All right. Good afternoon, everyone. I'm Dedipya Pasupuleti. I'm from Mercer University, Atlanta, and I will be your moderator for this webinar, which is organized by AAPS, Pharmaceutical Development and Preclinical Discovery PDPD community. This community focuses on the essential collaboration of scientific disciplines across drug discovery and early development, involving novel target and ligand exploration, property optimization, lead candidate selection, and effective transitioning of preclinical candidates into clinical assessment. Today's live event is titled Grad School to Entrepreneurship under the banner of Career Advice for Graduate and Postdoctoral Students. Our presenter today is Dr. Lindsay Pino. She's the co-founder and chief technology officer of Talis Bio. She leads the department in developing of Talis's screening platform, guiding and coordinating scientific efforts across automated proteomics, sample preparation, and high throughput mass spectrometry data acquisition and analysis. She has built and leads a team of multidisciplinary scientists encompassing the breadth of scientific work at Thales, including chemical biologists, protein chemists, and computational biologists. Lindsay received training from the Broad Institute, Pennsylvania, and the University of Washington. I am more than happy, and it's my pleasure to now present Dr. Pino for her talk. It's all yours.
Yeah. Good morning, everyone. And hopefully we can make this a little bit more of a discussion. I'm hoping that everyone's able to kind of walk away with both an idea of what might await you after school, after your training, and hopefully some inspiration on things that you might not have considered as options before. So this will be broken into roughly 3 parts on how you might take your research and what you're doing in your training right now and launching your own lab or your own company. A lot of the themes here are not just specific to entrepreneurship and launching a company as a startup, but can be relevant if you're going into academia and trying to start your own lab in a university. The first part is just going to be a little bit about my journey and how I got here. The second part will be how do you launch that lab? And then the third is just a little bit of insight into what the job looks like, which is surprisingly very little science. So career paths, as many of you have probably already experienced in your lives, don't typically start and finish the way you think they are. They take a lot of meandering paths that you might not have originally expected. That was certainly the case for me. So when I started out in undergrad, I was inspired by a book of poetry by a little boy, Matty Stepanek. He had a very rare disease called mitochondrial myopathy. And I found it fascinating and heartbreaking that we knew, as a scientific and a medical community, we knew exactly what was wrong with him. He had a dysfunction of his mitochondria, and he inherited those dysfunctional mitochondria from his mother. We knew exactly what was wrong, but we didn't know how to fix it. And that really inspired me to get into biochemistry and molecular biology as an undergrad. And as you can tell by this picture, my roommate took of me during a cram session for an organic chemistry test. I was not good at it. I really was not a great undergrad student and really struggled a lot. So when I was doing my undergrad research, when I was doing my undergrad studies, I thought maybe science is not really for me. So I graduated with my bachelor's degree and I moved abroad and I lived in South Korea for a couple of years where I taught English as a foreign language, TOEFL, to a group of high school students out in rural South Korea. I also worked with the South Korean Army, the Katusas there, who are ambassadors to the US. On kind of cultural and exchange English to help them build better relationships with the American soldiers that they collaborated with. And I really loved teaching. I found it very rewarding. when you're explaining something to a student and they just have that moment that they're like, oh my God, I finally get it. The difference between a and the, I finally understand. That was just very rewarding. But I really missed science. So I moved back to the States, to Boston, and spent a lot of time trying to find a job. So I got my undergrad degree. I graduated in 2009, which was not an amazing economic time to graduate, as I'm sure a lot of you are feeling right now in the biotech and economic world that we live in right now. So it was really tough. And it was hundreds of applications to jobs that, were not really what my dream job was, but it allowed me to initially get into the Broad Institute in the genomics platform, where I did a lot of kind of routine assembly line style sample preparation where thousands, tens of thousands, hundreds of thousands of samples. But the job was literally just reverse pipetting to check the volume, to see how much volume were in the submitted DNA samples. So hundreds, thousands, tens of thousands of samples, just reverse pipetting and recording the volume in the sample tubes. But I got my foot in the door at the Broad, and I was able to jump horizontally to a different platform, the proteomics platform. And there I got to do a little bit more research and development, not without many failures. If you've ever done any robotics or automation work, you might recognize to the right-hand side picture, the picture of a 96-well pipetting head of an assay map Bravo system, which I had crashed and broke that $10,000 pipetting head. So was maybe enjoying my job a little bit more, but realizing that there was a bit of a glass ceiling, and I might need to go get an advanced degree. So I decided to go to grad school, and that's really where I found my passion in mass spectrometry proteomics. And kind of during this time, mass spectrometry had both rose as a method of the year in 2012, and there was a lot of hype and excitement around mass spec proteomics, and then fell again. So mass spec proteomics as a technique, as a method, kind of overpromised and underdelivered as a whole. There was a lot of, we're going to do precision medicine, we're going to solve all these problems in biology that the Human Genome Project wasn't able to address, and then none of that actually happened. So the rise and fall was kind of happening. And I got to be kind of at the bleeding edge of some new techniques that were kind of revitalizing mass spec proteomics as a field. So being on the bleeding edge allowed me to kind of gamble a little bit on the skills that I was going to build in grad school. And I decided to build skills in coding, statistics, machine learning, because I was projecting, I was gambling that when proteomics was a little bit better, we were going to have a lot of data. And if we had a lot of data, we would need to figure out what to do with it. So if I tried to plot the field a couple moves ahead, 5 or 10 years ahead, I should learn how to do quantitative proteomics at scale and do machine learning. So that's what I did in that carving that very specific niche and making a name, a personal brand or a reputation around quantitative proteomics at scale opened up a lot of opportunities for me to teach other people how to do what I was learning how to do. So I got involved with a lot of workshops. I got to travel internationally to teach people, teach labs how to set up the systems that I was pioneering how to set up. Got to talk to a lot of pharmaceutical companies. And so it was around here, around this time, at the end of my PhD and into my postdoc, that I started realizing I'm teaching a lot of other people to do this. This might be something valuable that people care about. So maybe there's some opportunity here. And what that opportunity kind of came to fruition in the form of a collaboration that had happened when I was a grad student. And I had a lot of collaborations as grad students. I think a lot of PIs in grad school like to give collaborations to their students under the guise of, oh, it's low-hanging fruit. You'll get this easy paper, an easy middle authorship, co-authorship. It's almost never like that, right? You get a collaboration and it's like, a failed project that you're trying desperately to resurrect. But this particular collaboration ended up being amazing. My collaborator on this project was a postdoc at the time, and he had this really interesting way of separating out transcription factor proteins from binding to DNA. So we were able to look across different cells and see which transcription factors were binding to DNA in those cells. And really all he needed from me was a way to measure all of the proteins in those samples. And as I had just described, that was the niche that I had built for myself, quantitatively measuring proteins at scale and making sense of that data. So this collaboration was really cool. And he was just a really fun person to work with. If you have someone like that in your lab, in your department, in your network that you love collaborating with, that you're, working all day in the lab, and then you still want to go hang out with them afterwards because you really enjoy them, um, that's really important. That's really special. So we became good friends, good collaborators, and he eventually became my co-founder with this company. We published a paper from this collaboration describing this technique of purifying transcription factors off of DNA. Very academic technique, very manual, required centrifugation. But the paper came out and something interesting happened. So that's kind of the journey that led up to here. But this is kind of this first part of how do we get here? And maybe not a super linear path, but happy to take any questions if there was anything so far.
Lindsay, I have a question for you.
Yeah.
So I really enjoy the fact that you talk about nonlinearity in your path, because I think a lot of times, especially when I'm interacting with students, there is this idea that step one, you get an undergraduate degree, step two, you get a graduate degree, step three, you get a job, and it's this beautiful, seamless process. So what advice would you have for those that are currently in graduate school? And I think you hit the nail on the head with, like, right now being a very tumultuous time in the biopharmaceutical and biotherapeutical sector. What would your advice be to those people in terms of, patience, biding your time, knowing that there's not a perfect answer for everyone and it's going to be unique for everybody?
Yeah. I mean, everybody's situation is a little bit different. And, financially, there might be some pressures too. I was young, single, I didn't have a family, so that definitely made things easier for me to spend some time trying things that were maybe a little out of left field. But I do think it was very valuable for me getting to where I am now that I took I had the opportunity to try a lot of different things to really, one, cement my passion in this one particular niche. I was able to try some other things and realize, no, this is definitely what gets me excited to wake up in the morning and go to work and where like troubleshoot those 99% failure experiments and get through those for that 1%. So that I knew that I was all in on this type of science. And this was particularly the niche that I loved. I wouldn't have known that if I had gone straight from undergrad because I thought I was so bad at science at the time and I thought it was a personal problem, when really I just didn't like that type of science in that particular lab environment. And then I got to try teaching. I got to see like this is interesting, but maybe not something that's going to be make or break for me. So maybe tenure track where I would have to teach a lot. I would enjoy it, but maybe it's not actually that important and critical to my job satisfaction to have to teach. It's really the research and the problem solving I enjoy. So taking some time and uh, being a little bit humbled, that whole first job in industry was just checking volumes and putting stickers on things. And I only had a bachelor's degree at the time, so maybe that was all I was really qualified to do. But it also ended up being very valuable because I got to see how genomics was doing super large scale productionalized sample preparations. And now that's exactly what I'm helping set up at my company. Nobody has really done this for proteomics before, but because I've seen it in a sister technology, I kind of have that pattern to match. So it's super valuable if you can take that time or do that internship, take that like 1 year gap to try to do something that maybe feels a little bit crazy, then you'll never have the regret when you look back. You'll never think like, oh, but what if I had tried that one weird thing? You'll never have that regret. And I think that's super valuable.
Thank you. I appreciate that.
So, Lindsay, that was a very good presentation. Like, well, you are in the middle of presentation. You have not finished it yet. But
Yeah.
I was wondering, like, what are the enthusiasm points that give you the idea to go into like entrepreneurship?
Yes.
That is exactly I'm trying to. Maybe you are going to illustrate it later.
Yes.
But that is my point. I'm trying to figure it out. But your career track is really very interesting. That from undergraduate, how you went from doing the job in, like, say for example, teaching America, teaching English and all these things to getting into the graduate program. That is really very interesting. But I'm trying to figure it out. What was the like burning points that actually led you to those areas? Please
Yeah.
go ahead.
No, that's like this second whole section is just kind of tracking that because after that paper got published, that's kind of what sparked the next kind of section. But Shanghao, did you have a question?
Oh yeah, I appreciate that you're sharing about your how you graduated, then how you started your company. I was a grad student before, so I remember that time. So we did have some like a good science technology, and for students at that moment, I was wondering how we know that our technology we invented, we spend time on it, could be useful. And so,
Perfect. Perfect segue.
particularly for those PhD students, that is always challenging.
Yes, that's a fantastic segue to this second section. So we got this paper published, and one of the things that you'll have to consider is before you publish, you also need to make sure you have your intellectual property patented. So you can't just publish the paper and then do a patent. That makes it a little bit harder. So at the same time that this paper was being put together for publication, we were also filing for that patent. So this patent, through a very interesting route, the Altius Institute, my co-founder, the first author on the paper, was co-advised in his postdoc by the University of Washington and the Altius Institute. The Altius Institute actually is a kind of branch of GSK. So when the paper got published and we were filing the patent, GSK had the first right to this method. GSK said they didn't want it. So that meant this patent was out there, this paper was out there, the paper gets published, and all of a sudden we're getting all of this inbound interest. So at the same time that I'm getting all of this inbound interest from people to set up their quantitative proteomics and get their mass spec proteomics set up the way I had been working on in grad school, my co-founder is also getting all of this inbound interest from this paper from pharmaceutical and biotech companies saying this is a really interesting method. So then we have to decide, there is interest out there around this method. Maybe you've seen this interest because you've developed your technology or you've given this talk at a conference and you've just had industry people tell you like, wow, this is really interesting. Or you've got some molecule that you've gotten up, you've worked up some molecule, some asset to like mouse studies, and there's some really interesting PDPK on your molecule. You would go make sure you've protected that intellectual property, and then you gamble. So one of the first questions we were asking ourselves, are we even allowed to start a company? How do you even do this? And there's a really good article that I think is a great gut check from an accelerator program called Y Combinator. And it's specifically about how do you spin out your scientific research into a startup. So I'll summarize the article really briefly on the side here. First, it is really difficult to do a company while you keep your academic career. So if you want to be a postdoc and have a company, if you want to be a professor and have a company, that is incredibly difficult, because it means you'll only ever have 50% of your brain thinking about each one of those jobs. So it's a little bit, you have to pick. It's a fork in the road. Do you want to start this company, or do you want to pursue that academic career? Misconception number 2, you don't need a CEO. You can be the CEO. Scientist founders make fantastic CEOs. You don't need someone with business experience. So my co-founder and I are both technical founders. Neither of us has any formal training. We don't have MBAs. We don't have any business school experience. And fourth, you don't need to raise money first to leave the university. So I was still finishing my postdoc while we were getting the company together. And most universities will allow you to consult for a company up to some certain number of hours per week or per month. So I was able to consult for my company while we were getting all of these pieces together, while we were raising money, and then I was able to make the break and go full time on the company. So this might look, we were just talking about this a little bit, but the actual concrete steps, you need to figure out what that product is. Do you have some platform method? Do you have some small molecule asset? Do you have some particular chemistry or like synthetic route that you've helped invent? Lock down that IP. So if you're developing this in a university, you're going to go talk to your university's tech transfer office. They'll help you draw up all of that legal paperwork to file the patent, and work with you on licensing, giving you the license rights to your patent. You incorporate your company. Delaware C Corp is kind of the standard. It's pretty straightforward to do this, but your university will probably also have help for this. And then you start talking to people. Talk to as many people as you can about 3 main topics. The first is, who is important for your team? Can you do this by yourself? If not, who do you need to get on your team? 2, what are the scientific and technical milestones that you need to hit, and what are the business milestones? So for this fourth bit, the figuring out, defining, and figuring out what those milestones are, there's a lot of help out there. So if you have that scientific project idea, you can turn that into a business model using resources that are already available. So there are things called I-Corps. The NSF and NIH both have these innovation corporation innovation core programs. A lot of universities have these already embedded in them. So you can go talk to them and figure out, like, here's my idea, help me turn this idea into some business milestones. So you can do that. We used the NSF I-Corps here at University of Washington, which has a tech transfer office called CoMotion. A lot of universities have these. You just need to like look up your university name, tech transfer office, and you'll probably find it. And then just get ready for a lot of rejection. You really have to double down. Don't reject yourself. Don't tell yourself, oh, I'm not even going to try it. Plenty of other people are going to be more than happy to reject you. Don't reject yourself. Let them do it for you. Apply to everything and everything that you can. You never know which one of these shots is going to be a goal. So find incubators and accelerators. Your institute or your university almost certainly has an incubator or accelerator to spin out tech that's coming out of research in their labs. I just mentioned the NSF and NIH I-Corps. There are other accelerator programs: IndieBio, Y Combinator, Creative Destruction, BioFoundry. All 4 of these are competitive programs. You have to put in an application and apply to get in. But I don't think it ever costs anything to apply. So apply to everything. You never know when you're going to get into one. Doing as many of these as possible helps you get some of those advisors who know exactly what you need to do next. So that's what we did. We applied to almost all of these. We did the NSF NIH I-Corps, we did Y Combinator, we did Creative Destruction. All of these agencies support US startups. Several of these support non-US startups as well. I know Y Combinator and Creative Destruction have international presence as well. I'm not sure about IndieBio and BioFoundry, but all of these you can look up, especially if you're incorporating as a Delaware C Corp company. You have a U.S. presence there, so you can automatically kind of play that card. So that's what we did. So we applied to all of these. We raised that first round using a lot of advice that we got from doing the NSF I-Corps program, and then just going out and talking to a lot of people, hundreds of people, and having them reject us over and over again. But with each one of those rejections, getting feedback and incorporating some of that feedback, right? Like a lot of us are in science, we're used to 99% of our experiments failing. We're used to people picking apart our work that we poured our time and blood and energy and tears into. We're used to it. So it's just turning that side of your brain on and going out, getting a lot of feedback from a lot of these different places and incorporating that, getting better every time you get rejected. We found a little bit of space. So I had a small cell culture closet in Philly. It turns out you can just rent lab space, just like you rent an apartment. You can just rent lab space. So we had to do this because we didn't really have incubator space available to us from a university, but universities almost always have a whole set of labs that are specifically for technology that is being spun out of their academic labs. And a lot of universities will let you stay in your academic lab, especially if your PI is on your patent as well. If your PI is on your intellectual property, they'll let you just kind of like stick around and keep doing what you're doing until you're ready and have the funds to spin out. So it's one of those things, getting involved, getting connected with your tech transfer office. They're almost always more than enthusiastic to help you out. That was kind of like that first bit of some of the signals that we were getting that we had product market fit. We had, you know, industry and academics reaching out, wanting to learn from us what we were doing so that they could do it too. The fact that other people were interested in doing what we were doing was one of our very first signs that maybe we should just do it ourselves instead of teaching everybody else how to do it. Maybe we just do it ourselves and have this company. So any questions on kind of this part?
I have questions.
Yeah.
So it was, I mean, you answered my previous question very well. That was my kind of challenges at that time when I was a student. So now, let's say you have your idea, you feel confident it should be, you know, it's great technology. But when you really reach to the founder of other like funding agencies or other companies, as you mentioned, you will be rejected multiple times. Now, here's my question. In this process, it was not easy. How did you navigate yourself through this rejection? Do you feel really, do you have a point that you really feel, oh, why should I start with this?
Yeah, I will say resilience and figuring out where your stress releases are is really important. Both my co-founder and I do like to go for runs. So we like to run, and that's kind of our stress release. We go running um, and just making time every day or deliberately scheduling time right after you have some of those meetings to go for a run. It's one of the things that we did, especially early on. And just trying to take each one of those rejections as an opportunity to learn. If people tell you no, it's not that you have to take no as an answer or not take no as an answer, but just ask like, what is it you're looking for? Like, could you say a little bit more about what would get you excited, or what parts about this are red flags for you? And then each time someone's giving you that piece of information, we have like this huge notes document of why are they passing? Why are they saying no? What are they looking for? And then going back into the lab and doing the experiment that either proves them wrong. So now we can go back to them and be like, I love that feedback. We took that feedback and we did an experiment, and here's what we discovered. That has turned people from no's into yeses because they're, one, They like seeing that you're taking their feedback seriously, that you're not offended by being rejected. And 3, you are going to make it work. So that has been really helpful for us when we get feedback on our platform that just say like, oh, like you can only do 12 samples. Like we really want to do thousands of samples. Like, okay, let's go to work in the lab and figure out how do we scale from dozens of samples to thousands of samples. And then going back to them and say, hey, okay, now we've got 10,000 samples. What do you think now? Or going out and having people say like, okay, we want to see, data in a mouse. We want to see a program get into in vivo. It's like, all right, so we're going to go take some time, work up a molecule, do a mouse study, and then go back to them and say, hey, loved your feedback. We did a PDX study. Here's some efficacy data. Yeah, so trying to take it as an opportunity, not just a rejection, but an opportunity. And like a little bit of a gut check. I think sometimes, especially as scientists, we get stuck in rabbit holes. We think something's really cool and exciting, and then we go out and show the world, and the world's like, that is not cool or exciting. That's kind of weird.
So Lindsay, I actually have a question off that point. It's kind of a double-edged question.
Yeah.
When you're going out to these potential investors, first question is, what is the population like audience-wise? Is it a mix of businessmen? Is it a mix of science individuals? Is it more heavy to one side or the other? Does it depend on the funding agency? And then number 2, the last point you made is the second question I have is, how do you incorporate not just the coolness factor of your therapeutic or your technology, but also there's potential that this is going to make you a lot of money factor, and making sure that the business people capture that and see the value in it from an investment standpoint?
Yeah, all really good questions. Your pitch and how you're talking about your idea and your company should change for every different person you're talking to. So it's a lot of work. It's not just you have the same 10 slide deck and you go shop that to hundreds of people. Every time you're getting ready for a meeting, you try to look at who is on the other side of that meeting and tailor how you're presenting to that person's background, because there will be a mix of people. Some people will have very deep technical scientific backgrounds, but not in your specific area. So how do I turn my pitch for drugging transcription factors into something that a deeply intelligent and experienced neuroscientist is going to care about? Or how am I going to turn my pitch deck into something a medicinal chemist is going to care about versus that neuroscientist? So it's going to be a slightly different presentation every time you do it, depending on who's on the other side. And then you mentioned scientists versus business people. The type of investor that you talk to, there's generally speaking, you can lump investors into 2 buckets. There's the Boston bucket and there's the Silicon Valley bucket. So the Boston bucket is huge check sizes and tend to be kind of powerhouse, very high-powered companies and investors. The Boston money is like very deeply pharmaceutical focused, I would say. And then the Silicon Valley money tends to be smaller check sizes, but more deeply interested in technology. So we are, as a company, for our first couple rounds of fundraising, we are more that Silicon Valley bucket of money. We are a technology, a platform, an algorithm that we're developing. And as we mature as a company and have a deeper pipeline of programs, drug discovery programs and development programs, as that pipeline gets more mature and deeper, then the plan is that then we transition to those high-powered pharmaceutical Boston-based buckets of money that are looking more for assets and actual drugs. So it's kind of like these two different scenarios. So when we're pitching to these Silicon Valley people, we're really focused on the promise of the platform and the idea that, you know, if this platform works the way we're telling you, there's the long tail of here's how many potential diseases that we could cure that involve transcription factors. What is the market size of those cancers? How many patients per year have those cancers? Here's this like magic number as a platform. Other companies that have done these platform style business models. Here's that. When you talk to more the Boston based, they're looking for like those individual assets. Like, okay, we have a chordoma program, but the chordoma program could be secondary indication in breast cancer or prostate cancer or whatever, and talking more from that asset side. So yeah, it depends. depends on who you're talking to, and it's like kind of up to you as a founder or a prospective founder to do your homework on who's sitting on the other side of that call and what type of person are they. Do you put all of your slides that talk about the business and the potential payout and market? Do you put those at the beginning or do you put those at the end? If it's a business person, you put it at the beginning. But if it's a scientist, you want to talk about, you know, the platform and the science and why the science is going to work. And then
Great.
I saw a question in the chat
Yeah.
about coping with losses. I mean, coping with financial losses, you tighten your belt and try to come up with alternative ways of raising money. So financial losses, you can be venture capital backed. So we were just talking about investors, but there's also a lot of non-dilutive grants from federal agencies called SBIRs. So these small business grants, if you've ever written a fellowship or if you've ever written a grant, especially like an R21 style grant, these SBIRs are incredibly analogous to that, with the only slight difference being you need to like have a business justification. You can't just propose science for the sake of it. It has to be like very specific, like why does this help your company? Why do you need to do this scientific project? Why do you need to hit this milestone? And there's lots of webinars out there by NSF and NIH to describe the SBIR application process. And you can reach out directly, just like for a normal fellowship or an academic grant, you can reach out to those program officers and just chat with them, learn more about the process and how to do it. And if you're still in a university or an institute, their grants office can probably help you with that too.
So Lindsay, I have one question for you.
Yeah.
So among all these speeches you made, have you ever encountered a proposal from the businesses to be part of their entity, but rejecting to see you as your individual entity? And if you did, how did you face that?
Yeah, we have gotten maybe one or two offers to be acquired, but we are in this for, we're in it to win it. So those offers are in our general exit strategy plan, much lower than we want them to be. The goal, we really strongly feel like the technology and the platform that we have is going to be massively influential in discovering drugs for transcription factors. And kind of the dream is we're going to find privileged scaffolds in chemistry to target transcription factors, just like how for kinases, there's this privileged scaffold chemical like starting point. You can think of a privileged scaffold as being like a chemical starting point that if you want to drug a kinase, there's like kind of these general starting points that you can begin with and build out molecules that way. We don't have that for transcription factors yet, but that's one of the things that we think our platform could be used for. So we don't want to see our technology get bought by a big pharmaceutical company and then just get dusty on a shelf and nobody use it anymore. Because it's difficult, it's hard. If it was easy, someone would have already been doing it in a big pharmaceutical company. So we have just kindly rejected those advances and instead tried to turn that around and say, you know, we're not interested in selling the whole platform, but we be really interested in strategic partnership. If you've got some transcription factors that you want to drug, let's discuss going into a strategic partnership together, and we'll use our platform to find some hits for your transcription factors, or we can use your compound libraries to screen for transcription factor hits from your chemistry.
Yeah, thank you.
And part of that is just being like very aligned with your co-founder and eventually your board of directors on what the exit plan is. If the exit plan is to get out quick and potentially not really make a lot of money off of your exit, that's a perfectly valid thing, and plenty of people do that. But we're in it to really become a drug discovery engine for transcription factors. So yeah. But yeah, I've got this last little third, which maybe gets into a little bit what a day in the life looks like and kind of what's next. So I'll start off and just say I quantitatively work a lot more now than I did in grad school. I thought grad school would be the worst, but I have like a productivity like to-do app that I started using during grad school, and you can definitely see an uptick around the time that we launched the company in mid 2020. You can kind of start seeing it get a little crazy. That said, most of the work that I do is not actually science, so I can quantitatively break this down for you. I spend a lot of time in meetings. Sometimes these are investor meetings, sometimes these are board meetings, sometimes these are strategic partner meetings, these are meetings with my team, this is meetings with my co-founder. I spend a lot of time in meetings. A lot of time not spent in meetings is spent preparing for those meetings. So making slide decks, planning projects, making project updates. So trying to synthesize out of all the data that we're kind of generating in the lab. I had mentioned how not every person you talk to is going to be trained in your specific niche. So you need to figure out, how do you turn this data, this like highly technical proteomics data, how do I turn this into something a neuroscientist is going to understand? How do I turn this into something a medicinal chemist is going to care about? How do I turn this into something that an MBA, a business person, is going to resonate with? I do some other things, but the light blue bar is all the meetings. So I do a little bit of admin work. When you're a startup company, you have to do just kind of stupid things like the financial bookkeeping, so that you can file your taxes. You have to, like do payroll. You have to, approve PTO. You have all of your one-on-one meetings. You try to do a little bit of outreach. The vast majority of stuff that I'm doing that's not kind of research and turning data into these decks is called business development. And business development is really just money, right? So business development is putting together fundraising decks, putting together introductions and scheduling meetings with different venture capital investment firms, writing grants, putting together pitch decks to talk with pharmaceutical companies, managing a customer relation database. So keeping track of everybody we've talked to, when's the last time we talked to them, and when we might, like, what is the thing, why did they reject us, right? And you just have a giant spreadsheet, one for investors, one for pharmaceutical companies, and just keeping a running tally of everything that they didn't like about you, so that when you have solved that thing they didn't like, you can go back to that sheet and say, this person wanted mouse data and I have mouse data now. I should go let them know, like, hey, it's been 6 months since we talked last time. I'd love to give you an update on our latest mouse study. Or, this other person loved what we're doing, but they're not interested in oncology. They're interested in cardiovascular disease or interested in obesity and diabetes. Like, all right, maybe we'll do some preliminary experiments to prove some feasibility in these other indications. And then I'll go back to my sheet and say, oh, these 5 people liked us, but didn't like the indication data we had. Let's go revisit those conversations now that we have a little bit of data in cardiomyocytes or something. So most of my job is doing that. So now I have a small team, and the scientists do the science, and I spend a lot of my time trying to translate the science that's being done to our board of directors, to these business style people, to these scientists in other areas like the medicinal chemists, so that they can make use of our data. And then they circle back to me and let me know their follow-up questions. They'll ask questions, and now we got to get back in the lab to generate more data to answer those questions. So in like kind of the life cycle of a startup company like mine, which is more this drug discovery platform, not a single asset company, but a drug discovery platform. The first stage of your company, you raise 1, maybe 2 million, something around that, to start your lab. So this is very similar to an academic startup, right? In an academic startup, maybe you get like $1 million or so in a startup package to start your lab, and that's supposed to last you a couple years to get your equipment, get your lab set up, build a small team of trainees, and get some of those first papers and grants. Exact same thing in the startup world. You get like $1 million or so. You could apply for grants to get more, but you get $1 million or so to build a team, build your lab, and just prove that your idea has any traction. Does anybody care about your business idea? And then you need to prove that it's technically possible. You've proved now that it's theoretically possible, that making this company is a potential thing. For us, that meant we had to turn our centrifugation manual protocol into something that was 96 well plate automated. So that was our first, like, is it even theoretically possible to turn this approach into a drug discovery engine? And then there's this, is it technically possible? Can we actually make a small molecule therapeutic? After this, we're going to be asked to do it again. They're going to say, that's great. You did it once. You have 1 drug. Now make 2 or 3 drugs. And then finally, the last stage will be, did that drug, did those programs make it through to a clinical, commercialized drug? So that'll be, like a good 7, 8 years before we have something that is that far down the pipeline, and will mean we need to have, like 30 potential programs going to prove that it's scalable and repeatable, and make it to that commercially viable. We're going to need, a good 30 programs, because 29 of them are going to fail at some point. Either preclinical, they're going to fail, phase 1, they're going to fail, phase 2, they're going to fail, phase 3, they're going to fail somewhere. But one of them is going to make it to that commercially viable stage. And so that's where we kind of are now. We just raised that 10 million. We've got this productional drug discovery platform going. Our challenge now is to come up with a deep set of programs. We have a lead program in mice for drugging the brachyury transcription factor. Now we need to come up with, a handful more transcription factors with hits, and start building those out so that we can start pushing towards proving that what we're doing is scalable and repeatable. So that's where we're at. The last thing that I'm going to mention is network always. Network as much as you can. Almost everyone on my team has come either through a network recommendation, a friend, a colleague that I've worked with in the past that have said, hey, you should hire this person, they're fantastic, or I directly worked with them, like my co-founder and I. So you never really know. The person sitting next to you, the person working next to you at the bench could get you your next job. You never really know. So keeping in touch with some of those networks, just spending, an hour or so a month to reach out to people on LinkedIn. Hey, it's been a while. Like, saw that you're working at AstraZeneca now. Congrats. Hope things are going well. And just keeping those contacts up to date and refreshed. So that is everything that I wanted to kind of cover, like what led up to starting this company, what those early first steps were in starting the company, and what it looks like now, a couple years in. Happy to take more questions.
Thank you, Lindsay.
Yeah. Oh, yeah. Go ahead.
No, go ahead, Shang. I was just going to announce, like, if there are any questions, feel free to shoot out.
Okay, I may start. So when we are looking out where we have the project, we are looking for money.
Yes.
especially for the angel investor, and you can ask the money based on your budget, right? Or, I mean, sometimes we don't really know how much budget we should put there. So my question is, as for those angel investor, what quantity of amount, what amount of money is reasonable to ask for them? I cannot, based on my project, they say I cannot ask them for 4 million, 5 million, right? But if you ask them 200,000, probably they will say, oh, it's too less. But what would be the right number to start? Obviously, depends on your project. Let's say drug discovery, early discovery stage. And yeah, that's my question. It's a situation.
Yeah. I mean, it looks totally different for every specific business and every specific stage of the business. Really, yeah, there's so many factors. So really, before you start with the budget, I would say start with what are you trying to do? Do you literally have nothing? Like what is it you need to do first? And what that milestone is, and then work backwards from what that milestone is, because that will dictate what your budget is and how much money you're asking for in that angel, like friends and family round. You want to start with something. So like if you're building some device, you want a prototype. Like step one is just, do you have a prototype? And working backwards from there. Okay, well, to get a prototype, first I have to, like find a manufacturer to build my prototype, get some quotes on that to find out how much that's going to cost. Can you do it by yourself? Or do you need someone else? Okay, factor in, you need to build out a team and how much that's going to cost. Do you have a place to do it? Do you need rent? Do you need to build some lab space? Some rough costs. So get some quotes on like going online, going around your town, going to your university or your institute and asking, how much would it cost per month for me to rent a bench? What equipment are you going to need? Are you going to need to buy some equipment, or is your PI or your institute going to loan that to you? So working backwards from whatever that is. If it's, I need to make, I need a drug with, like, I've got a starting point that looks good in mouse, like, okay, well, what is the next thing that you need? Do you need to get all the way to a DC? Do you need IND? Like, what exactly? Do you just need a lead? What step do you need? And then working backwards, okay, just like an estimate, what do you need? Like, you've got mouse and now you need some CRO work to do ADME, do PDPK, like you need however many rounds of medchem. Who's going to do that? Do you do it? Do you go into the lab and synthesize that? Lab space, starting materials, equipment. Are you going to contract that out and use another company? Okay, get some quotes for some potential, and then working backwards from whatever that budget is and figuring out what that milestone is. Just talk to as many people as possible. Like, what would make you believe this is a viable business and a potential business, and just getting their feedback. And you'll get a lot of ranges. You'll get people that say, like, you need to have human data for your asset. And you're going to be like, yeah, that's not feasible. We're not going to do this by myself. And you'll get a range from, like, you don't need anything. You can do it right now. Great. Go talk to potential investors. And if they say no, then you weren't actually ready to go do it right now. So yeah, I would say start with, what is the milestone? What do you need to achieve? And then work backwards from that.
So just a follow-up question on that. Do you advise that a prototype should always be ready before you approach an investor? Because sometimes just to make that prototype once you're out of the university is very hard. Like, you need the investments, and you need the money to rent the bench, or as the space that is needed. So I mean, I don't know, like, are there any investors out there who would actually invest in making the prototype itself, or no?
Yeah, I mean, like, there are some very early stage investors and angel networks that would do those very early safes, uncapped safe. So instead of doing a priced round, you do an uncapped safe round. And there's a lot of these accelerator programs that are focused specifically on that stage where you've got maybe a really good idea. Maybe you had like a working, like janky, you soldered it together yourself prototype, and now you're looking for like a slightly sleeker potential prototype. There's people that are looking exactly for that. So, and you only find out who those right people are by talking to as many people as possible. And a lot of people will tell you like, I'm not the right person, but I'll refer you, like I'll forward your email to my friend, and my friend might know the right person. And then you just keep getting daisy chained down that until you find the right person. So if you get that no, and you get that like, we don't invest in this, just be like, thanks so much for your time. Do you happen to know anybody who might be interested in this? And asking for that next referral, one, saves time on your part, and two, then you have that network effect, right? Because now for every one no you get, if you ask like, do you have a friend or two, and now you get 2 leads, and then those 2 leads turn into 4 leads, and you can start following that down to find who is the right person. And there's grants, so you could always apply for grants as well.
Okay.
Yeah.
So Lindsay, could you please tell us your experience about SBIR grants?
Yeah, I find SBIRs fantastic. So similar to academic grants, NSF is more kind of the basic research. NIH is more specific to human health. Look for specific special, what are they called? The special announcements with specific calls. I can't remember what they're called, but because there's the omnibus, and that's just kind of like the global like catch-all for any grant. But then there are also these special interests, special calls. And like a year ago, for example, there was a special call for alternative protein sequencing. Like if you had a method and you just didn't have like a prototype yet, that would have been the perfect grant for you, because they were specifically looking to fund prototyping technologies that would be alternative protein sequencers. Or maybe they're looking specifically for pain management. Do you have any approaches, whether it's like an app or a drug that would help with pain management? There's specific things for that. So looking for those RFAs and applying to those, getting to know the program officers. They're different program officers. The SBIRs have different program officers even within the same institute. So if you're like National Cancer Institute, SBIR is a different program officer than like the academic grants. So you want to make sure you're talking to the right person. If you're still a postdoc and you're transitioning out, there are transition grants, just like there are K99s to transition from a postdoc into an academic PI-ship. There are similar transition grants for transitioning a postdoc into an entrepreneur. So looking for those special announcements as well can be really helpful. And then same thing there, prepare for rejection.
So do you have to have like established lab and spaces and everything to apply for the SBIR?
No, you just need to have an incorporated company. You need to like file your government like small business number. But the grant application tells you exactly what those identifier numbers you need. So you just have to be incorporated and like have some preliminary data. Like if it's strictly just a theoretical idea, you might have trouble getting funded because there's no evidence that it's going to work. But if you have some preliminary data, you really don't need anything. You can put all that into your budget. You can put into your budget, you get your 40% or whatever overhead, have your rent, have your salary, have your equipment, whatever you need.
Got it. Thank you.
Yeah.
And this preliminary data can be any published data as well, right? As long as
published data,
you're the author.
Yeah, published data is totally fine. You want to make sure you have like some evidence of like, do you have the intellectual property? If it's just out there, can someone else just steal that and do it? So you want to have like some legal protections around the intellectual property, and you can work with your institute to try to get those. Because if you developed the technology, the prototype, the method, the device, the molecule, if you developed that while you were training at an institute, they, I think, like legally own your research. So you'll want to make sure that they're involved because they have very deep pockets and can lawyer you much easily than you can defend yourself.
Right.
Yeah.
All right. Any more questions? All right. So I see that our time is just about up, and I don't see any more hands raising. So I would like to thank our presenter, Dr. Lindsay Pino, for her excellent presentation. And on behalf of PDPD community, I thank you for all of your participation. Also, in approximately 1 week, today's webinar will be available for replay on the AAPS e-learning website. So you may now disconnect from the webinar, but you